And greetings, everybody. Welcome to the Evidence and Reasons for the Christian Faith video channel. I'm Salvador Cordova, and I'm very privileged tonight to be joined by my longtime friend, Jonathan Bartlett. So for some of those who are new to the channel, and we keep growing our subscriber base, thank God, I'm an independent molecular biophysics researcher. I worked for world-famous geneticist John C. Sanford for 7 years while I took care of my elderly and disabled mother for 7 years, and I got to work from home, and I took care of her for 18 years total. And goodness, Jonathan has met my mother. That that time he got stuck in DC, and
Yes.
had to spend the night at my house.
Sal had to drag me to the airport at 4:30 in the morning. That was quite the adventure.
Oh, that was fun. So he and I go a long ways back, and so this channel, it tends to cater to Christians who are also graduate students in sciences. My background is physics and biology. I have 5 science degrees. Although I'm being challenged now because I'm entering nuclear physics, which I'm not that familiar with, even though I have a physics degree. Nuclear was not my specialty, and Mr. Jetty and Jordan K. and John B., another John B., who's a paleogeochemist, certainly stretching my knowledge in these areas, and they are offering very credible challenges to the young Earth model. And I mean, I sincerely say that. I try to entertain the opposing viewpoints because I've said the young Earth model is not out of the woods yet. It has a lot of difficulties. This is in contrast, I mean, the young Earth as far as the youth of the universe and the Earth. But as far as intelligent design and arguments against naturalistic abiogenesis, arguments against the evolution of complexity, like say eukaryotic evolution, I think the young Earth creationists and the old Earth creationists definitely hold the high ground there, and they're not going to stop. We're gonna see this avalanche of data coming in that evolutionary biology cannot explain. They might say, it's common descent. We can even accept that for the sake of argument. But even assuming common descent, it's becoming evident there has to be miracles. And I know some people who watch this will vociferously disagree, but I've debated that topic. But in contrast, stuff with radiometric dating and other things with the young Earth creationism, I actually have a softer stance from a scientific standpoint. I accept it by faith. I am a young Earth creationist. I'm a young cosmos creationist, but I cannot defend that really on empirical and theoretical grounds as easily as I would the other things with evolutionary theory. And so tonight we're going to have a nerd session that starts to go back to more of my bread and butter, where I got paid, which was in biology, even though I love physics. And even though I no longer work as a paid employee at Dr. Sanford, I still work with him. We're trying to publish papers together, and hopefully even more this year. We got in secular venues last year, and also with Joe DeWeese. And I got to publish in Jonathan Bartlett's Blythe Institute Communications, and I'm going to give him a chance to introduce himself shortly. But tonight we're going to have a nerd torture session. That's this channel is becoming infamous or famous for that, where we really get into professional level creationism and intelligent design research here. And this isn't just like an advocacy channel. This is not an advocacy channel. This has been a chance to fellowship and also to just show what it's like to be a professional researcher in creationism. And so with that, Jonathan, feel free to introduce yourself, and you could talk about Blythe Institute, the CELS, and the Adaptive Systems Group, and the Discovery Institute. Have at it.
Yeah. So thanks for that intro. So yeah, my name is Jonathan Bartlett, and so I've been doing independent research in the intelligent design and creation movements since about 2005. And when I originally got into it, it was mostly for apologetics. And but as I started getting into it, I realized that there was actually a lot that was there that of practical value. That there was a lot of things that science actually. It wasn't so much that uh, uh, Christians need science to verify things in Scripture, but that science was actually hurting itself because it needed ideas from Christianity. And I realized that there was a lot of things that were a lot of problems with biological models that you really needed to have an outside perspective to help see. And that's kind of what I kind of had a shift and started working more in the practical side of intelligent design and creation. So one of the things that I've done is um, I published a series of papers on how to measure the amount of information that a cell contributes to its own evolution. So this is something that is um, it's something that you basically wouldn't think of as a Darwinian evolutionist, because you usually think about information being added to the genome and not the genome itself having information to do evolution. And so, for example, if you have something that adapts to something, if it's able to adapt easily, well, you say, well, why is it able to adapt easily? Is it able to adapt easily because the adaptation itself is easy, or is it able to adapt easily because there's there's information in there that predisposes it to being able to adapt quickly? Um, and so, are there sensors? Are there triggers? What all is going on that is helping organisms figure out how to adapt to their environment? And that turns out to be an absolutely fascinating question that it seems that it's hard for the other side to even pose the question well because of their assumptions coming in. And it kind of it gets in the way of them being able to really ask deep questions about what's even going on in biology. And so that's kind of been my focus for the last 10 to 15 years. I've uh, I've been doing publications with biocomplexity and communications of the Blythe Institute, some older ones in Creation Research Society and um, the occasional papers of the BSG, just on what what are the practical aspects of recognizing that information doesn't just arise out of nothing, that actually requires some amount of design to get into it. So that
In
that's been my focus.
Well, thank you. And just so you know, and also the audience, I have to run all these controls. And if you see me just scurrying back and forth, I'm not trying to be rude to my guest, but usually some of the more professional shows will have someone like, what was her name? Cheshire Vic handling all the controls, like on the Non Sequitur show. And
Yep.
it's great when you have someone handling all that. I have to do that. And so if you're seeing me scurrying up, trying to, handle the side chat, and it's occasionally we get bot attacks and I have to just handle all the controls. So I want to say greetings to everyone here. Patrick Alexander, Redeemed SWCM, and Bent Hovind. Bent was here yesterday. He's no relation to Kent, and we had an interesting talk. Bent is a Christian brother. He's a theistic evolutionist. Yeah, Sal got a haircut. Yeah. So tonight's going to be a nerd torture session. We are, like I said, we showcase professional creationism here. This is not popular level creationism. This is definitely not Kent Hovindism or any of the, like the 3-paragraph papers that you'll see to try to, advance creationist theory. That's just not going to cut it for those especially who are going off into graduate school, who actually, John and I actually talk to groups of professors and stuff. So, oh, aloha, greetings. So we have a variety of topics. you mentioned adaptation. Last year, John Sanford, myself, and others, Ola Hosder and lead author Bill Basner, we published in a secular venue, Springer Nature reference work that actually was very critical of the definition of adaptation in evolutionary biology. We got an earful from the critics, and I said, well, what did we do wrong? We just quoted the top tier like Joe Felsenstein and Richard Lewontin and Grodwall, and there are all these problems here that you're not acknowledging. If adaptation means the fittest, the most reproductively successful are the ones that end up reproducing the most, I'm just like, well, that's kind of tautologous. In mathematics and logic, we call that an analytic statement. If and only if. It's kind of it's a tautology. And that's okay in mathematics, but if you're trying to formulate scientific theories, you have to create synthetic theories. So like a tautology is saying being deaf is not hearing, or a synthetic statement is your eardrum is working, therefore you're going to be deaf. That's a synthetic statement. And the problem with the definition of adaptation that evolutionary biologists use, it's absolutely useless to explain what they really want, which is in Origin of Species, Chapter 6, the organs of extreme perfection and complication. And it's like, you can't use the definition of adaptation that the population geneticist adopted, where the most fit is the one that reproduces the most, because it's decoupled from complexity. It's a disaster. And people,
Well,
go ahead, John.
What I've found is that a lot of people will, there's kind of, in many areas, there's kind of two sets of definitions. And one of them is the one that's more well recognized with lay people, and another set of definitions is the one that's used in more technical literature. And oftentimes what happens is that if you use a definition that's the popular one, they'll criticize you for not using the more technical one. And if you use the more technical one, they'll say, well, that's not the definition that's in the textbooks. And and so I think that's kind of like, you're quoting the experts. Well, and so if you criticize the expert version of it, then they're going to say, okay, well, you didn't attack the main definition. But if you would have attacked the main definition, they would have said, oh, well, you didn't address what the top experts say about it. And that's kind of what I found in a lot of things, is that there's kind of like there's a lot of different definitions for different terms. And if people want to criticize you, they'll kind of they'll pick one definition and just excoriate you for not following that particular definition, even though there might be several equally valid definitions of a word. I've seen that happen. I've been on the receiving end of that many times myself.
Well, fundamentally, this whole thing at adaptation is better expressed in terms of engineering principles. And what I mean by that is, I just maybe by example rather than giving formalism here, because it just confused matters. Greetings, Janet. God bless you, dear sister. And by the way, JohnnyB you are a lay Christian minister. Is that fair? We will offer prayers for people if I remember to do this toward the end or somewhere here. I'm going to invite JohnnyB to pray for us, for our community. We're becoming a tight-knit group here. So the engineering definition is like, okay, an airplane is adapted, well adapted. It's suitable to operate in the air. Spaceship is suitably constructed to operate in space. A submarine will not operate on land. It's suitable to operate undersea. And like, we don't have to use reproductive success to define quote unquote adaptation in that sense. And if you think about it, that's exactly like, okay, so a bat is very suitable to operate in a cave where there's no light. It has a sonar system. You don't have to, my jaw just drops when they say, oh, it's adapted because that's the one that's most reproductively successful. I'm just like, an engineer would just like kind of puke.
Yeah.
Like, what do you mean? It has sonar. That's what makes it able to operate in a dark
The
environment. Come on.
yeah the internal feedback controls, I find something that gets oftentimes overlooked. Um, just the fact that um, these things, there's a very narrow range of feedback controls that actually work for anything. And so when you have these complicated systems, well, there's not a selectable pathway, because there's selection actually is actually pointing the wrong way throughout much of the journey. And that actually, I have a paper under review about this, but that's actually a necessary fact of any um, any complicated system. You actually have to have uh, areas that you would traverse that where selection is naturally going to be pointing the wrong way. And it's kind of a complicated reason, but it actually falls back to um, the halting problem and Gödel incompleteness theorems that show that uh, for any non-trivial function, you actually have to traverse space where selection is going to point the wrong way.
Amazing. Now, Janet is, she's a dear sister. She had lost her husband some years ago, and she has some disability, and she's very close to my heart because I took care of my mother, and she had a similar situation. I was wondering, Johnny B, if you could pray for Janet right now and the gang here before we move on to this technical discussion, and also you could pray for our time here together. I'd like to do this
Yes,
publicly.
absolutely. Lord, we just thank you. Lord, we pray especially for Janet during this time, and Lord, I just pray for comfort for her. And Lord, I pray for peace. Um, and Lord, I just pray for all the people who are watching. Lord, I just pray that you would raise up this generation in knowledge and understanding and wisdom. And Lord, that we would, um, Lord, that we would be your lights in the world. And Lord, just help us to give glory to you in everything that we do. Help us to get closer to truth, um, and just lead us in the right way. And Lord, we just thank you for everything that you've given us. We thank you for this amazing world, and we thank you for each other. In the name of Jesus, we pray. Amen.
Amen. Amen. So again, this is going to be nerd torture, but we're trying to destroy evolutionary theory here because we think it's wrong. What I mean by destroy, I think it's already self-destructed, but we're just trying to highlight that because it's destroying people's faith. And by evolutionary theory, not so much the common descent. I'm going to grant that for the sake of argument. It's the evolution of complexity, the organs of extreme perfection and complication. And that's the thing that you know, evolutionary biologists will conflate common descent with evolutionary complexity. And I can have a more formal presentation on that, where there is an important distinction, particularly with orphan proteins. And I don't want to, well, actually, we'll probably touch on that tonight because I was asked to develop this talk of developing the tools to analyze engineering systems. But the point is as we analyze as an engineering system and we start taking inventory of the parts, we can do kind of as Johnny B here has said, we may not have to use Turing's and Gödel's incompleteness theorem. We can use a lot of common sense to say this is natural selection is not going to work this out. The other important thing is we can actually build, in at least in principle, experiments that'll show natural selection is not going to work. I'll give you an easy one. My distinguished colleague, adjunct professor at Vanderbilt and associate professor at Freed-Hardeman, Joe DeWeese, he gave a great talk. I don't know if you're there, Johnny B, or at least saw it on topoisomerase, and the topoisomerase video came
Saw
out.
part of that. Yeah.
Okay, here's a testable principle. Put a partially formed topoisomerase in a cell. See if it even lives. Okay, how how can you select something that's dead? And try to just throw out any other pathway. I'll give you any pathway you think you can start off with, initial conditions. And I will predict you're not going to, let's say this is a testable prediction. If you keep saying, well, it wasn't a fully formed topoisomerase, some other pathway. It's like, okay, here's a testable prediction, experimental prediction. You're not going to find one that's partially formed, and you're not going to find selection pressures that are going to form it. That's a testable prediction. It's falsifiable. And I don't think you're going to succeed. And if that's the case, definitely then evolutionary theory is on very shaky ground for that just one protein. And there are thousands of them here. And we're going to, JohnnyB asked me to make a presentation to kind of teach this to his group that's affiliated. I don't know all the mechanics of it, with the Discovery Institute. So maybe this is a good opportunity. You and I maybe you can describe what the Discovery Institute is in relation to creationism. I have my own definitions. And then specifically the CELS and then the Adaptive Systems Group. Okay, if you could.
Yeah.
So here.
Yeah. So the Discovery Institute. So they are. They do a lot of funding and promotion of intelligent design and related fields. And so the Discovery Institute. They have a couple different centers. The Center for Science and Culture is largely does intelligent design related to biology and cosmology. And basically what they're doing is they're looking at the world in terms of are there features that reflect actions of agents, because we know that we know what it looks like for something to be creative, and so the question is, are there things in the world that look like they were the results of creativity? And so that's kind of what that center does. And they have another center called the Center for Natural and Artificial Intelligence, and I'm part of that. And
I did not know that. That's a new center.
Yeah. So
wow.
The Center for Natural and Artificial Intelligence that focuses on machine learning, artificial intelligence, and it's actually really intelligent design applied to computer science. And actually, for my church, I actually have a series of kind of practical theology talks that I give, and one of them is the first one is called how to cut a cake using the doctrine of sin, and the second one is how to make cheese using the doctrine of creation, and the third one is how to build an artificial intelligence using the doctrine of man. And so anyway, and if you think I'm being cheeky or metaphorical, I'm actually not. All three of those are precisely what we cover. But basically, the idea is that a lot of the things that we do in practice actually have their roots in theology. And one of the things that most people don't understand is there's a lot that goes into computer science and artificial intelligence that the best artificial intelligence systems actually take into account the nature of humans from a theological standpoint. So, for example, if you think about Google, what makes Google so good? And what makes Google so good is they do what I like to call intentionality harvesting. Everything that they do goes towards figuring out how to better recognize what the intentions of creative people are, and then record that and kind of play it back to you later. So if you think about it, when back before Google, for those of you who were online before Google, all the search engines were just text matches. So you just typed in something, and it would do text match across the entire internet and just pulled back whatever had the best matching text. But the problem is that people would just add in all sorts of keywords at the bottom of their pages. And yeah, someone said Alta Vista. Yeah, absolutely. Just put keywords all over the bottom of their pages, and so they would get matched for all sorts of search keywords. And so what Google figured out is that what people were really intentional about were the links. That if my page linked to yours, that was a huge mark of intentionality, especially the word that I used to link to you and the fact that I linked to you. That was it was marking much more of an intention of the page creator. And so Google's algorithm made use of the fact that people had more intentionality when they were creating links than when they were creating text. They went further because when people click on links in the search engine, they can actually tell that somebody's being, if this link is being clicked on a lot more than this other link, well, people are being intentional about clicking on that link so that it's more important. So they learn to harvest intentionality in people. And all of the best AI systems learn to harvest intentionality of humans, or they actually just use humans as fake computers. That's called artificial intelligence. And so anyway, basically understanding that there is a divide between what humans can do and what machines can do, and figuring out how to put which process with which group is a great engineering problem, but it integrates some theological understandings with it.
I didn't even know American Online Brave existed, so thank you for that, Michelle. I wanted to say, okay, so we were talking about the part of the Discovery Institute. They actually had projects in transportation, democracy, and technology. Their transportation stuff was in Seattle. It's funded by Bill Gates, the Bill and Melinda Gates Foundation. Even though my understanding is sadly Bill and Melinda are splitting, the foundation goes on. We could probably better characterize the Discovery Institute as it's a secular institution. The people who promote intelligent design there are by and large, there's some notable exceptions, they're by and large old Earth creationists or old Earth ID proponents like Michael Behe, where they accept common descent.
Yeah, Michael Behe would, I mean, he probably wouldn't like the label theistic evolution, but
Correct.
he is a old Earth intelligent design who accepts common descent.
Common descent. Stephen Meyer, I had an interview with him. He doesn't talk about this much. He describes himself as, you could argue that he's a progressive creationist. So using this is very inexact, but to say the Discovery Institute, I'd say is an old Earth creationist outfit, but they're secular. They're secular. That means there are no professions of faith. One reason this is important, you have a lot of people that are Jewish. You have people of other faiths. You even have, the ID tradition has had both pagan and atheist contributions. The most notable atheist would be Fred Hoyle. The former atheist would be Frank Tipler, and there's some others. Any advocates of extraterrestrials who are atheists would be considered ID advocates at the biological level. So the importance of it being secular is it makes the argument much more forceful. We will argue, the tradition had been to argue purely on scientific grounds about criticizing evolutionary theory and saying it doesn't agree with what we know about physics, chemistry, cellular biology, etc. You can't make those assertions. You can't say that it's natural. You have to actually prove the mechanism. And they've been very, very good at that. They're just exceptional at that. Now, anyone wants to see an intelligent design-like argument prosecuted, it'd be Joe DeWeese, Topoi-Somres, the way everything you said would fit pretty much in a secular classroom, and it's still a devastating critique of Darwinism from a scientific standpoint, not a theological. Young Earth creationism, on the other hand, invokes a lot of, creationism is a mix of creation science, creation theology. It has a lot of theology in it. So just as an aside, John and I had the privilege of being there in Dallas, and I was at the Institute of Creation Research the day before, and then we hung out at Dallas. And I don't know if Matt's is here tonight. We hung out.
Yeah,
Go on,
I keep
John.
on losing track because you asked me to talk about cells and ERG as well, and I got lost talking about the Center for Natural and Artificial Intelligence. But yeah, so we were at the Center for Science and Culture's Dallas Dallas
Faith and
Conference on
science.
Science and Faith. Yeah,
Yeah.
and hooked up there. But Discovery also has they've been putting together what's called the Engineering Research Group. And so what the Engineering Research Group is it's been basically the idea is that the people in this group kind of believe that the ID has won the argument technically, and so we're kind of essentially we're kind of over arguing about it, and our goal is to simply move forward in practical applications and research
Right.
and show how does engineering how does if we say that biology is design systems, how does um, how can we take that? Well, if they're designed, that means they're engineered, and we have engineering professions, so that means that engineers might have something to say about how are these things put together. They might have some insights on how these different systems work together,
And
and
why
so
they were adapted to the environment.
exactly. And so we've got a couple different teams. There's like a control systems team.
Wow.
There's a sensing team. And then I'm running the adaptive systems team, which is looking into how do organisms change and adapt.
Now, I know this may seem kind of like far-fetched, but I'll give you a tangible example. I was studying cellular neuroscience at the NIH FAES graduate school. I was taught by one of their top researchers, and the research team that he headed, and he had colleagues there. It was great. And one of the assistant professors he recruited had no formal biology training. He had a PhD in electrical engineering. And why would we think that? If you actually look and see if I could show a picture of this. For one thing, the ear processes audio signals. And that has been the staple of engineering, electrical engineering, for a long time. And I'm sorry to be insulting here. What good is an evolutionary biologist going to be to help you understand how to process signals? It's all about transduction of signals. It's right in there. The people with the right mathematical background to understand how the ear works would be an engineer. And obviously, we're trying to diagnose problems in hearing. So this whole thing, it's like if you want to understand bat sonar, you want to get an engineer who actually probably worked on sonar systems. What good is getting an evolutionary biologist and all he knows how to do is build phylogenetic diagrams that are probably wrong anyway? Sorry to be insulting, but that's, you could tell where my biases are. So now we're actually applying the design paradigm. You're getting the specialties and the knowledge, because I've been appalled at some of the things I've heard from kind of the evolutionary community. And it's like if you had a little bit background in Shannon information theory and it's Shannon's theorems, you wouldn't be saying some of the stuff you say.
Mm-hmm.
You're like, well, why does DNA need error correction? Why won't the designer build it right the first place? Well, why do you think we build compact disks with error correction? It's because the most compact form of creating storage is to have it stored wrong and then to correct it later. That's the most optimal way if your design goal is for compactness. You don't make it right the first time. And if you'd actually studied Shannon's theorem instead of evolutionary theory, you'd know that. So
Right.
sorry to be on my soapbox,
I,
exercised
I,
here.
I remember you did a great article on that on Uncommon Descent many eons ago.
I've said, thank you for remembering. I'm going to show ear neuron diagram schematic. Now we're going to see if Google can give me the thing I'm looking for. Hang on, and I'm going to show it here for the benefit of, because when I was studying cellular neuroscience, I'm going to try to, I'm just going to try to illustrate your point here. And so we had to study this. And Ben Rex, if you're listening, he's our resident electrical engineer. And he and I have some kinship because that's my background too before I got into biology. But you're like, well, and really at the beginning of the creation evolution debate, it was always the engineers against evolutionary biologists. And they were always saying, you guys are irrelevant. You don't know anything. And that's changing, because what do you see on research teams? You see, I would bet there are probably a larger proportion of engineers, computer scientists, physicists in medical research than there are evolutionary biologists. Certainly, I'll see these teams, and I was asking, I mean, you just have to go to some of these research teams that study cellular neuroscience. Look at their qualifications. Most of them are biochemists, and you might get, like, say, an electrical engineer. So here is a diagram of how we actually represent the electronics in a neuron, in a nerve cell. It looks shockingly similar to the circuits we electrical engineers work with. We just use different substrates, but it's very much the same principles. Some evolutionary biologist that all he knows how to do is phylogenetic diagrams, it's not going to understand the complexity of this and characterize it correctly. If he's going to try to define adaptation in terms of reproductive success, when this is what you really need, he'll be sort of laughed at. And that's why, when I see the job openings for people that do this. So I'm just trying to kind of reinforce your point there, JohnnyB, about the importance
if anyone
of
in
the
the
ERT
chat,
group.
it looks like I'm frozen. Can you hear me?
I can hear you, Johnny.
Okay. For some reason, my video froze.
It'll happen a little bit on Restream. That's what you get. You get what you pay for, which is the free service.
Sounds good. So, yeah, if anyone in the chat is interested in learning how to read schematics like those, I actually have a book out called Electronics for Beginners. So you can find that on Amazon. It's a little
You're a man of many
plug.
talents.
So
by the way, I need to promote. I'm sorry to go on a rabbit trail, but when we hang out, we just love hanging out. We haven't done this. We didn't have enough time down there in Dallas to hang out. But JohnnyB published in peer-reviewed. He corrected notation in mathematics. He's also a calculus teacher. I got to just rave on that because that was a huge contribution. So JohnnyB and I love talking about calculus. I mean, this is scary, Johnny. On a Friday night, two guys hanging out and talking about Leibniz notation.
So yeah, if anyone wants to see my calculus contribution, the paper is called "Extending the Algebraic Manipulation of Differentials," algebraic manipulability of differentials, and basically it's about the fact that, just to get super nerdy, in calculus you've got the first derivative and you've got the second derivative, and what they always tell you is that you shouldn't, even though the second derivative is written like a fraction, you shouldn't use it as a fraction. But the reason for that is actually because we're writing it wrong, and if you write it correctly, then you can use it as a fraction. And so anyway, and so that's why I figured out how to write it correctly so that we could use it as a fraction.
So congratulations on getting published in a secular journal. And so, it just feels good, Johnny, to be published in the secular world. And So congratulations on that. And I think that was a great development in mathematics. So thank you very much for your contribution there. I just had to just laud the accomplishment, this
Thank you.
out of the box thinking. So this is a preamble to everything we're actually going to try to do tonight. And I was supposed to give a tutorial, but I feel it was important to give a kind of a background of why we're going to cover some of the topics that we have here tonight. And so what I had on the docket, it's almost daunting, and I doubt we're going to get onto it. But there's so many databases and tools like Uniprot, SPARCLE, BLAST, CDART, CDD, Mega, Muscle, ENCODE, PFAM, PhosphoSite, PISA, Pymol, GenBank, RefSeq, Hidden Markov, Gene Prediction, PacBio, Illumina, Sanger, DCA, RScape, AlphaFold 2, etc. I don't know if we're going to get to all that, but these are the tools that will help us prosecute our case for design. These are some of the tools, and this will be a chance to maybe cover them. So maybe the best way to go about this, and I invited John here because he wanted me to give a presentation to the Discovery Institute ERG group, and since he leads the Adaptive Systems group, I figured he could give me feedback as to what to include and maybe what to exclude in the presentation. So let me bring up a paper that I'm working on, and we're going to submit it to for peer review. Now, the peer-reviewed system has changed a little bit. People have preprints because they actually want to get a little bit of feedback before they submit to the editor. So it used to be all this very secretive process. So depending on what you're doing, they're bypassing that now to get a better, freer flow of information, and also to confirm that you are the author, because there have been some scary stories where the reviewers kind of just lift the data, like, oh, I didn't know that project was going on, and then so you lose priority. So
Well, and on top of that, the time for publication is just can get ridiculous. I mean, a lot of those things have a lot of the journals have 6-month review cycles, and
like longer.
yeah, it's just like I can't hardly believe that people can make progress with those kind of delays. And it's not it'd be one thing if there really was like 6 months worth of work happening, in those review processes. But usually by the time the paper is submitted, the majority of the work is done, and this is mostly just paper pushing. So it's just really sad to see that that it just takes so much effort to get anything out. And anyway, that's my personal annoyance with the peer review system. There's a couple journals who do it well, but
Right. So
yeah,
really, all this
I
to
think
say, yeah, go on.
I was gonna say I think there was a year on my calculus paper between submitting it and it getting accepted, and there wasn't more than maybe 4 paragraphs worth of changes in the whole thing. So it's not like there was an intensive back and forth with the reviewers or anything like that. It was just it was pretty much just waiting on people for 6 months.
So the point of all me mentioning that is that this is also a chance for me to get some review before the submission, and so there's I'm finding this is actually a better process because I've actually gotten some very good corrective feedback. So I'm just going to start with the basics. Some of this, John, you already know, but since we have an audience and this is good practice, and then also, I don't, there might be some people who might join the talk that, they could use a little refresher on this. So what I like to do is just say, okay, we have a protein here like collagen. And let me see if I can get this in a different, I don't know why let me just try this one instead. See if this works. Okay, that works. So we have a protein here like collagen, and let me try to make this even a little bigger. It's a known protein. A lot of our body is made of collagen. The proteins, that is, 20 to 30%. You can get this collagen at Walmart. It's a beauty product. Collagen's a protein. It's made of amino acids. We can use English alphabetic letters to describe each amino acid. And so then you can also describe the protein sequence in terms, it'll look like an English rendering. This is the, I'm going to expand this. This is the alpha, the collagen 1 alpha 1 paralog in humans. And let me expand it more so you could see the lettering. And there's actually a non-random pattern here. This is about 1,400 amino acids or residues. And so let me try to show you can actually see it right there. Definitely a non-random pattern. And this is going to be important as we talk about, I'm going to refer to the collagens to talk about the databases. And so you can see this non-random pattern here. And I'm going to show some other proteins. There's that every third residue, there's a section of it that's a glycine, a G. And now this is a human zinc finger protein. It's 540 amino acids, zinc finger 136. I'm going to expand it, and there's also a non-random pattern here. Very important. The C's are cysteines, and the H's are histidines. That's where the zinc ion will actually connect. Each row is what we would call a zinc finger or zinc finger domain. It has a separate isolated modular fold. And this is also important in understanding the databases, because then you begin to see, like a lot of engineering things, the modularity in construction. And definitely you can kind of sense the modularity in construction here. And as I add more layers of coloring, you'll actually start to see this. And there's a tool called CDART that will actually help you see this modularity. Now, it's not the best database. So sometimes when you have to go to individual peer-reviewed papers, you'll actually get the breakdown where they will start to highlight the parts. And with this Venn diagram here, we can group together some of the amino acids to be certain classes. And when you do that, you'll even get more patterns here to talk about that. Now, the collagen and zinc fingers are exceptional in that you could visually see these patterns. For most proteins we will actually deal with, you will not see, it will not be this easy. You'd have to be some sort of savant to see the pattern. And so you could definitely see, just as an aside, you could see why common descent is going to be problematic. There's not going to be any gene that's going to be ancestor to all these without making major, you can have like little changes within species, but they're definitely different architectures. Like there's different architectures for pianos, blenders, land vehicles, and helicopters. There are variations within that class, but you're not going to evolve one from the other. It just doesn't make sense. You can definitely see that at the protein level.
The other part of that is that it has to, all of these things require architectures to work, as does the gene. And it's, so not only can you not switch architectures, selection kind of requires an architecture to already be there for it to work. And and so that's the deeper thing that I think a lot of people just don't think about, is that the architecture has to already be there for selection to have anything to work with at all.
And you could click, and I'm happy to argue with evolutionary biologists over just the two proteins, and I have, and I don't think they won that exchange. I'm just, in fact, the way they argued with me actually showed that they didn't understand the mechanics of this. And one thing you could see is natural selection would actually resist the evolution of one form to the other. because
Mhm.
before you make a good collagen to make it into a zinc finger, it's going to be a bad collagen, or vice
Okay.
versa. And they'll say, well, no evolutionary biologist is saying that. And I'll say, okay, we'll grant that. But I'm highlighting the point, you're going to have similar problems if you try to synthesize a consensus common ancestor for both. So right there, you're starting to have the orchard model, which the creationists would absolutely love for the major protein architectures. It starts to look Linnaean. A phylet, you can't use, you would be very limited in trying to apply phylogenetics at the major level. You can at the lower level. You get some very, you can build, you definitely have hierarchies, but the hierarchies don't have one single ancestor. You can't have one universal phylogeny. So it is an orchard. An orchard does say that there is phylogeny within a class. And the databases will help you. The databases will help you refute universal common ancestry at the family level. You're going to get orchards, but then the databases will also help you identify the functional components. And so if we're talking about adaptation, since you're doing the adaptation point, we'll build on that with some of the other databases. If you have, just stop me if you have a
Yeah.
question, John. This is for your benefit. Oh, so here's an example where you can build a phylogeny. So the human zinc finger, and I use Captain Kirk as representative of the human race. And you have a pig. They have 50 to 60% sequence similarity, and they're different scoring methods. Depending on which scoring method you use, you'll get a slightly different figure. But you could see that it has the same architecture. So it's like, okay, this is the orchard. Definitely you could say this looks like common descent, but I showed you where it doesn't, where it fails. It's like trying to look at an orchard and make a universal tree when really the orchard's the better model. And then you can build this phylogenetic tree for that individual tree in the orchard. And we could do the same for collagen. And there's on the left is the human collagen, and on the right is the zebrafish. 75% similarity, same architecture. So again, this is reinforcing the orchard model. We're not saying that you can't build a phylogenetic tree, but you can't make one that's a universal tree. It will be an orchard, and that I think that really will reflect also at the organismal level. So this is hope that the orchard model is going to rule someday if people are open. So that's again alluding to that. So now we're going to talk then about. I'm going to skip a little bit ahead. This is a publication that Joe DeWeese and I did, and this is going to relate to a very important database called Uniprot. But when we're talking about adaptation, let me see if I could zoom this up a little bit so it's a little clearer. Yeah, see there, that was our publication in a secular peer-reviewed work.
Very nice.
And yeah, oh, praise God for that. And this is going to show the complexity of the protein, and I think we've talked about that before. And this is just a segment of the topoisomerase 2 protein. We have the 2 alpha paralog in humans, which is this sequence here, and the 2 beta. And this is just the C-terminal domain, which is like the last 100 or so residues, amino acids. Each of the arrows shows the modifications, chemical modifications we can make to each of the amino acids. We can add a phosphorus group, a phosphate group in purple. The orange is a sumolation group. There's a chemical term, I'm not saying that quite right, but it's a the chemical modification is called a sumolation, and that's an acronym for something I can't remember. The blue is for a methylation, the green is for an acetylation, and there's one where you can have even 2 different possible kinds, a ubiquination or a sumolation. And so if this looks like a computer punch card, that was kind of intentional. So you could see here the other kinds of creatures here that have also the topoisomerase 2 in them. And there's a database for these modifications. Does that kind of resemble to you like changing bits in a protein? The protein is like a random access memory.
And it can take changes for to set states.
Yes, and it does two things. This is like unlike any kind of engineering we do, because in a chip you just modify it, you don't change the shape of the chip. Here you flip a bit, it actually also will change the conformation. It'll subtly change the shape of the protein, and this ends up then becoming like a Swiss pocket knife. You can reconfigure it to adapt to different multi-uses. So this isn't adaptation at the organismal level. You have adaptation right down to the individual protein level. We're rather confident, I'm personally confident, that the switches on and off relate to the cell type that the topoisomerase is expressed in, and probably the cell phase.
Yeah,
And
that's one thing that I think has had a lot too little research on, is how genes and expressions and proteins, how they differ in different cell types, because it seems like there's I mean, I think most people recognize that they do different things, slightly differently in different cell types, but it's never taken quite as seriously as it seems to need to be.
Right. And that's why I think our work is important. We're trying to raise awareness even within the ID, and I hope you don't mind me calling you out as a creationist. It's outing you as a creationist.
I call myself a creationist. The one thing that I do note, I do try to tell people, is that, like, I believe in creation. I believe in intelligent design. I try to be explicit about what anytime I make an argument or a paper or anything like that, I try to be explicit about what my inputs are and what my logical basis is. So if I'm arguing about if my basis is scripture, I will say so. If my basis is physics, I'll say that. If my basis is computer science or engineering principles, I'll say that. So that's kind of I just try to always be specific about what it is that I'm arguing from, so people know what the foundation is.
Right. And I think it's important to argue from the secular perspective, like in Acts 17 in the Bible, because you get an independent witness from nature. You don't have to start with a Christian worldview to see that it's right. Because if we had to start with the Christian worldview to see that the Christian worldview is right, I think a lot of us wouldn't ever become Christian. So it's really good to be able to start from something that even people who are non-Christians will agree is true, and scientific data like this is an example. So I think it's great that you and I can start to highlight this, because when I've interacted with evolutionary biologists, I'm just like, did you even account for stuff like this? How can you prosecute what you're trying to do? And I'm like, well, actually, you can prosecute it better when you're actually ignorant of the facts, because if you actually had to account for this, I think your theory would start to fall apart very, very quickly. They resent me saying that, but this is I'm normally a very nice guy to the opposing side when we're discussing, but when it comes to just the hard facts, I got to be a little bit more forceful. It's like, well, being nice or being mean is irrelevant. It's just the facts here. The theory is going to fall apart, because each of these chemical modifications that we're seeing here, you need machinery for each of these. You need a feedback control system that's going to go and say, oh, we're in this cell type, in this condition, we're going to compute this, we're going to flip this bit, now it's in that state. And by the way, that's what we call a write operation. the all sorts of proteins that may bind or interact, proteins, glycans, who knows what, lipids, that interact with this, those are readers. So you have readers and writers, and you have, so in the case of the phosphate groups, the writer is called a kinase, the readers are the binding proteins, and the erasers are the phosphatases. So you have these machineries there, and it's not just sufficient to have a machinery that can read, write, and erase. You need a system that's going to control this, and that's just for one bit, one lousy bit in one protein.
Yeah.
And they're like, particularly in human eukaryotic cells, multi-cells, you have way more of these modifications than you do in a bacteria or yeast. I looked at the corresponding ones, and again, we're going to go to the databases. You will see that, evolutionary biologists will say, it's not like a progression from simple to complex, it's a radiation. I'm like, oh no. If you take proteins across species, you go from like bacteria, the bacterial corresponding thing, topoisomerase, which is the gyrase. You go to yeast, which is here. Actually, we have the yeast here. And then you go all the way to humans. You'll see going from 0 to like on the order of 50 to 100 post-translational modifications. There is a big jump in complexity in the way this thing works. One interesting thing is you can take human topoisomerase alpha and topoisomerase beta. We have 2 genes for each of the 2 classes of topoisomerase. You could put it in a yeast, the yeast will be fine with either one. It's happy with it. You could the yeast can function with a yeast topoisomerase, a human alpha 2B, or a human top 2 alpha, or human top 2 beta, or a yeast top 2. Doesn't matter. You can't do the first. We have experimental evidence for that. Now, ethically, we can't do that to live human re-engineer. We did it with something close, with mice. If we knocked out the alpha, the mice dies. If we knock out the beta, the mice dies. It needs both simultaneously. So we learned something. We are more complex. It's not just radiation. There is a staircase toward complexity. And it's just nice to be able to back it up with data. And so how did I construct this diagram and all these other diagrams? That I wanted to show the diagrams first, and then we can go to the databases where I gleaned this from, because that was painful. If you have any comments or questions. Do you think this is helpful to make the presentation when I just start like this?
Yeah, I think looking at what visualizations are available is good.
Okay. All right, so now let's do the painful stuff. Let me take this down, and then I'm going to show then the Uniprot database. And so we're just going to, since we had topoisomerase there, I'll show you the Uniprot database. This is probably the most important, because then you can break things down. Were you at my, did you get to see my presentation that I made to the ERG group on?
I did not get to see that.
Okay, so I'll give you a little bit of this, and this is going to be cool, because as we go, if you look at the Uniprot database, it's like ho-hum, but when you know what it signifies, it's a different story. So so we looked at the topoisomerase, and this is probably I would say this is the central place you want to go if you're studying individual proteins.
Okay.
Now they're the genetic guys that study DNA. I study amino acids. Okay, so this is going to be amino acid heavy. They're people that do the genetic side, and it was kind of tough for me to work for John because he likes DNA, and I'm like, I like amino acids. So so one of the first challenges is to know the name of your protein and how it's stored, and that can be a nasty pain. And serendipity, I hate to say it, but sometimes you need a little serendipity to run into the right people to give you the right identifier. Once you have the right identifier, you can start. And that's a never-ending source of frustration. It's like, how can I, I want to look at this system and this protein. Sometimes you just can't type in the common name. You have to know the identifier. So that's kind of, that's going to be a tough lesson to learn. And I don't know any way around it. So that's why it's good we have a community at the Discovery Institute where we have people like Joe DeWeese, we can ask them. So once he's the one who told me use top 2A. Okay. So once you have the identifier, you can bring up this page and it has all this data that's collected from numerous sources for you. So let me just expand this so everyone can see it. So all I did is I went to the Uniprot website, and the website's called Uniprot, and I typed in the word top 2A, and that gave me a list of all the ones in the Uniprot database from all the species, all the paralogs, etc. I clicked on one of them, and this is the topoisomerase 2 alpha. And it's going to give you a ton of data on it. So the first thing, it'll identify okay, let's just go here, the sites, the major sites, the binding sites, the metal binding. And so now, what does that have to do with anything? Well, let me bring this up. This is from our paper here. One of the versions of our paper. Let's see if I can bring it up. See if this is the best one too. Okay, here's a good diagram. So how did I get all the colorings of this topoisomerase data? I went prot.
Okay.
That gives like the binding sites. So one of the binding sites for ATP is at position 91, the other at 120, and then there's this magnesium one at position 461. And I don't remember how I exactly did this, but this was painful because you could see that I don't have any numbering for the color, for the positions. So at position 461, it says DQ, DQ here. I think I had to figure a way to count that and identify it. Yeah, now I remember. I had to use some tricks to identify that that was the sequence in position 461. So position 461, it says DQ, DQ, like I call it Dairy Queen, Dairy Queen. Joe DeWeese's PhD dissertation dealt with those 4 letters. I'm pretty sure if he knocked out the D's, D stands for aspartic acid, I think. Correct me if someone out there, correct me if I'm wrong. The magnesium will not bind to it. The topoisomerase will fail. That's an example of a single critical point of failure. It's gone. And that was his PhD dissertation. Could you kill it? And there are other regions here that if you kill them, it's instant death for the topoisomerase. It has to be physically in the right location and the right letter. And so, when I showed this to John, he was really impressed. I said, yeah, once you make it visual, because when you have it just kind of laid out here, you're like, oh, that's kind of ho-hum. But when you actually see all the compact functionality, so then you have these regions here that Uniprot will identify in terms of modularity. And these were, again, I translated it into the coloring. And then they have all the links to where you could find the peer-reviewed literature that says this, and all the experiments that deduced it. This was painful.
So did you do the coloring yourself on those?
Yes.
Okay.
There's not any, because we can't make a general coloring scheme, because every protein has, some proteins don't have the, ATP binding sites or magnesium binding sites. So it tends to be just kind of on each, on a case-by-case basis. And this definitely blows away common ancestry for one single protein. You're just not going to be able to unify them because they're so different. This is why I love this Uniprot database, is if you can just kind of colorize the amino acids in your mind, you start to see how different they each are and how complex, and how you can't evolve one from the other. So I was just going to town on this database, and if you look at it, it's like, well, can't make any sense. How is this relevant to God and ID? And it's like, well, that's what we're here for. That's why they pay us the big bucks to do this, right? To teach it and to make the connections and how it points to intelligent design, and for the theists out there, how it points to God. And so,
So one thing, I don't know if you would know this, because one of the things that we're looking at is uh, we're looking at simple sequence repeats, and I don't know, are there any annotations that say, hey, this is a simple sequence repeat? This is if we're trying to find things that where so for those of you out there, simple sequence repeats are basically usually anywhere between 1 and 30 nucleotides that get repeated over and over again. And what makes them really interesting is that the simple sequence repeats, um, they are, first of all, they're the most mutable part of the genome, and they the most likely change to the simple sequence repeats are copy number variants. So it's through a process called slip strand mispairing, where the repeated sequence will pop in or pop out uh, multiple a different number of times when the DNA is copied. And so what we've been looking at is this is not our hypothesis originally, but we've been looking at in a little bit more depth than other people have, the idea that these are actually tuning knobs in the genome. And so we're kind of we've been trying to come up with various ways on how we might do the analysis to try to, because the question is, are there environmental variables where the environmental variable is tracked by a repeat length in uh, in the genome, and so that's kind of what what we're looking at in the question that we're asking.
oh my goodness, now I'm scared because I have no answer for that. And that is
So
not, you're going into DNA and I'm doing amino acids. So, okay, this is why we had this rehearsal, because this might be a shock,
yeah,
because what your group might be really looking for isn't what I'm giving. I'm hoping you're at
well,
least
yeah, no,
edified by our discussion tonight, but no,
yeah,
I don't.
absolutely. I mean, we're kind of like the tools. We're still pretty new at the tool, so that's why um, you know um, we're kind
Uh,
of at the
right.
beginning of our journey on that. So
I will tell you something interesting, since you're talking about repeats. Um, it's not a database or a tool. Google helped me find a paper. So for muscular dystrophy, the repeat number is very important. Are you familiar with that?
I no, not in particular.
Okay, it's cool and it's also disturbing. If um, I'm going to mess this up, so can you give me a moment to uh, Google
Yeah.
this and muscular dystrophy repeat? There's a particular there is a particular class of muscular dystrophy, um, repeat chromatin. Oh, here it is. FSHD. D4Z4 repeat. Okay, and I don't know. I'm having lots of Joe Biden moments this and being forgetful. If I remember to put this in the video description, I will. So this is very interesting. We have this thing called a D4Z4 repeat. It is 1,000. It's a kilobase or so, or 3 kilobases. And so you could see it's each of these where it says PCG. Well, anyway, it's a motif, D4Z4. If you have 11 to 100 of them, you're healthy. If you have between 1 to 11, you have muscular dystrophy. If you have 0, you have no muscular dystrophy. So either 0 or 11 and greater, you're fine. And the repeat number varies among individuals. But for some reason, when you get below that, it goes berserk. And one reason there is, you could probably, I don't know if that, I mean, going back to like, if we even, this is going a little bit into the metaphysical here, where we would hypothesize kind of like an intentional design. Did it even have that? Did D4Z4 even exist was the right design to have 0? And I don't know. Or was it to have something, that's above 11?
Right.
But, all of our medical practice does go to some Platonic archetype of what's right. I mean, can't run away from
honestly, that. You can't have a hospital without having an archetype.
Right. Exactly. The evolutionary biologists don't like that because that does suggest design, but it's like, well, operationally, from engineering and medical standpoint, you have archetypes. Exactly. So, but what it may do is it lengthens the DNA, and that changes how all the machinery can interact with it. So I'm almost guessing that if you just shut down the system, sometimes the default, if the system's just totally shut down, quote unquote amputated, then you're able to minimize the damage. That's kind of my idea, because the D4Z4 repeat is used to recruit. It's a docking station for molecular machines. So the repeats may enrich the number of machines, the concentration of them, and in and of itself, that could be important. That you can, for example, in with transcription factors, if you want to have more of the same transcription factor docking there and maybe doing a little bit more regulation, that's possibly one way you can increase the concentration. And so this illustrates a couple points how the repeat number can be variable, but this is definitely a case in point where the repeat number, if it's not right, it could lead
problematic.
To very problematic. So I'm sorry that, yeah.
So a question just in general, what is the benefits of using Uniprot versus NCBI or something else for doing these sorts of lookups? How is Uniprot different from?
It is a good starting point because it links to NCBI and BLAST and all the other tools. So it's an aggregator site. So if you have a known protein, you can go there. The problem is it's obviously protein based. So a lot of the things you're describing are in the non-coding regions. So, okay, what's the limitation? Oh, the coding region's what, 2% of the genome. So all the non-coding stuff, you're not going to get it through Uniprot.
Yeah, although a lot of the repeats we're looking at are coding region repeats.
I didn't, yeah. And then you have the inexact repeats, like what I was showing, like with the zinc finger, you could definitely see the repeats in the rows.
Mm-hmm.
And those are actually the repeats in the coding regions are interesting because you'll find the college, well, actually it's more subtle than that. Because of the codon degeneracy, you would actually not notice the repeat for what it means. Because when I was showing college, see if I have a collagen picture here. Sorry, I'm just going to whine a little bit. We have these guys in hot rods with their loud cars. It's just annoying. I can hear it. I don't know if you can hear it, but it's just, like, do these guys have anything better to do on a Friday night? It just sounds like artillery shells out there. They love having all the backfire. Anyway, remember, I'm highlighting all the G's here, but you could actually see all the different different codons that implement it. So if it's a different color, it's a different codon, even though it codes to glycine, which is actually an interesting pattern in and of itself. And so actually, what you just said is news to me. I didn't know that there are repeats, so many repeats within within the coding regions. Yeah.
So one of the ones we've been looking at is, and this was one that's interested to me because so in Arctic cod, there's a repeat element that's the antifreeze protein.
Mm-hmm.
And so it's interesting because, you know, the evolutionary biologists have assumed that that it's basically a repeat that got recruited into a gene, and you know there's some evidence for that. If you actually read the papers, they're like, oh, and then magically it got, you know, promoters attached, and you're like, okay, but you put that aside. It's interesting that there's a repeat expansion, and as and we haven't seen if this is, you know, if this correlation holds up, but it looks like as the fish are in colder waters, the repeat expands, and so the fish that live in colder areas have a bigger and bigger repeat, and that's actually also true of an entirely different antifreeze protein that has a different repeat. And if you and that one is I believe it's like a whole like 31 amino acid repeat.
Wow.
And basically so the amino acid goes in a circle, and basically it takes I think it takes 31 amino acids to make a complete circle, and so that whole amino acid will repeat. Whole amino sequence will repeat. As it does, the whole protein gets bigger and increases its antifreeze properties. And so it's interesting that the location of the repeat is precisely at the point where making the repeat affects it affects the environment variable directly, right? There's a correlation between the place where it is expanding the repeat and the environment variable that it's trying to track.
Right.
And so this is kind of this is the kind of thing that we're kind of looking at. The hard part just comes into trying to you know, go from just kind of looking at some genes and saying, oh, this looks like it's in colder water fish, and actually trying to, you know, say, you know, once you try to say, okay, well, is there like a linear relationship between how cold these fish can live and the size of the repeat? Well, you'd have to, you know, purchase yourself a nice little biochemistry lab to answer those questions. So anyway, that's kind of where we sit at the moment.
Okay, maybe I could help a little bit with that. I'll just make a kind of just a comment on the ideas. especially now that you and I know Joe DeWeese and I've worked with him. He studied topoisomerase for 17 years, and he has colleagues that their whole professional life was topoisomerase. What I'm finding is that even one protein is so hard to comprehend. It's very hard to generalize. We may learn from one protein to another. So a repeat in one protein may mean a totally different thing in another, and it's going to be hard to generalize it. I'm finding that, there's some rote generalizations we can make about protein probabilities and, this idea or that, but there's some things that are just so unique to them that you could be like Joe and just study it for 17 years and probably the rest of his professional life, and that's just one protein in one organism. So going back to the Uniprot, how this might help you at all is maybe it might be able to give you context of where the repeat is. Now I'm going to show you a more, you saw the collagen. When I take the Uniprot data and colorize it all, this is what you will get. It is just, to me, this is just mind-blowing. First off, the green location, the little green dots here, and I'm sorry it's hard to see, those are the post-translational modifications on the prolines. You need a special machinery to put it there. If it looks irregularly spaced, it's important. It's a design feature. It gives it the biophysical properties. If you don't have it there, you don't get the right kind of turning of the helix, because it would tend to form, you need to form something other than the alpha helix. So this is a situation, you have a chicken and egg paradox. You need to have this, you need to have another enzyme there to make this happen. You also need the collagen to have the motif that will draw it to make that post-translational modification. Then we have little parts of this where you have the postal code, the localization codes, and I'll cover that a little. Did I ever show you a video on the localization code?
I knew that there's something to the localization codes, but I don't know any details, and I've never seen the video on it.
That was in the ERG presentation that you were not able to attend, but I'll give you pieces of this, because once you start to know the pieces, then you might know what can actually repeat and not repeat. Because if you start repeating some of these things here, it's going to be pretty bad. Like, you can't just take random. So maybe the main contribution I can give here is you can identify that if it repeated here, it's going to be bad. It's going to be a disaster. But over here, the repeat is acceptable. So maybe Uniprot can help you identify that for this particular cod protein. And again, this is collagen. This isn't the cod. This isn't the antifreeze protein. But just to show the complexity, this whole first part here, up here, it'll be cleaved off. It'll be cut and becomes kind of a protein of its own. And then same for the bottom part. It'll be cleaved off. So you basically make one protein, it functions as one protein, and then it breaks apart to become 3 proteins. This is just nuts. A lot of foresight in that. And so they're able to identify these components here.
I see the people in the lab trying to figure that out for the first time. Can you imagine?
Oh my goodness.
What that would have been like? It would have been like, you have this one protein, and then all of a sudden you're like, you have 3 different ones. You're like, what happened to our old protein? And
okay, and so a little bit of chemistry
I just can't imagine
here.
being the first person.
Oh no, this is just brutal. And I thank God that it's studied. I mean, kind of in a sad way, it's to cure human diseases. That's why they're throwing money at this, and we have money to actually do all these experiments. And like I said, you can imagine Joe DeWeese just looking at one residue in the topoisomerase. That was his PhD dissertation for that aspartic acid in position 461. It's like, okay, Joe, this is your next 4 years. This is your life. And this is probably going to be informative here, just to give you a little bit of chemistry. So the C here is a cysteine. And so it'll connect to another cysteine via a sulfur. you create these sulfur bridges that affects the way it's shaped. So you definitely don't want to be folding this stuff. I mean, you don't want to be making repeats willy-nilly here. If you have sulfur bridges or salt bridges, sulfur bridges, or disulfide bonds, you'll hear that term. Michael Behe's paper with Dave Snow talked about the difficulty of evolving even one disulfide bond. And so if you wanted to make an argument that that this is targeted mutation, what will probably help is if you can build a diagram like that using Uniprot. Now, unfortunately, some proteins are much better characterized than others. Humans obviously have the best characterized proteins, select proteins. I don't know how much we can get on your cod protein, because you can see it's a lot of money just to be able to identify the sections. But I would look there. Cool. So that's the benefit of Uniprot. If you go to the non-coding sections, that's way outside of my expertise. That goes into the ENCODE territory. And I don't know, Jeff Tompkins is the only one that I know that knows how to navigate the ENCODE database. I don't know if Rob Carter, Jeff among our group is the only one that among our colleagues that has actually known how to touch the ENCODE database, because I don't know how to do it. And he knows how to do the gene browsing, and you could find repeats galore. So, so Uniprot's this start. Why don't I just, we're at 90 minutes, and before I totally wear you out, I'll try to navigate to the rest of the Uniprot database. So you can see all the hyperlinks. It's just great to give you all this data. You can see the localization in the cell. Goodness, look at all the chemistry databases. Yeah, this is a one-stop shop. So when I showed you all that diagram with all the post-translational modifications, that's only some of them. You can look at the list. This is the list of all the post-translational modifications. And I spent 6 weeks colorizing, because there's so many, and trying to understand what they meant. And there has to be like a motif there. So if you do just a willy-nilly repeat, you're going to break the post-translational modifications. So that'll probably strengthen your case if you find any. And maybe if you know the name of the protein, we can kind of guess and look in Uniprot. But let me just finish the one with the topoisomerase. And this is just, look at all those bits you're flipping. So the protein it has its function. It's also a random access memory. It's just unbelievable. Look at that. Look at that long list of positions.
Is this top 2A or is this top? Is that the one you're looking at right now?
Yep. And so it might even be just this from here to here was the diagram you saw that was all colorized with the arrows. But I had to make other diagrams for the other parts here. And you could see, and you just think, wow, that's just incredible.
So are the modified residues, does that mean post-translational modification?
Yep.
Gotcha.
Post-translational and the class and the position. And we haven't even decoded what they do. We have some suspicions. We know you could probably knock some of these out, and it's going to cause probably damage to a particular cell type. That's just my guess. And we actually, the reason Joe and I did a paper on this was we were suggesting that this could be a therapeutic target for chemotherapy. It's like, why don't we target the post-translational modifications rather than knocking out the whole topoisomerase? That might be, right now, it's kind of a shotgun where you just go after every cell with a chemotherapy, and hence when you're trying to kill a cancer cell, you end up killing lots of healthy cells. So we were suggesting that maybe we could target the regions of where the post-translational modifications are. We could have very targeted chemotherapies. Okay, so let me just, let me then expand this. You talked about how it links to the NCBI. Well, see, there's like their database is all over the place. It's just unbelievable. So there are these, they have these proteomic databases, the X-ray crystallography, if you wanted to see the shape of the topoisomerase. This is why the Uniprot's just a beautiful one-stop shop. All the positions where you have interactions with other molecules. You can imagine just the experimental community that had to deduce this. And this is only 1,500 amino acids.
Yeah, I can't imagine the number of people and time and energy and money that it took to do all this is just amazing.
I know. And like the evolutionary biologists will say, yeah, definitely involved. It's like you don't even know half the story. You don't even know 1% of the story. You're going to make all these assertions without any proof. You have to account for all this. Oh, okay, here are the protein data bank. Even Joe, this is, if I can brag on our colleague Joe, he was on the research teams. Some of the researchers contributed to this data that you're seeing here. The protein data bank has X-ray crystallography data. So that's how we're able to get these 3D images, these synthetic 3D images. And then we had the domains and oh, look, domains and repeats. Okay, I did not know that. So there's not a repeat here, but there might be one when you find your whatchamacallit, your antifreeze protein. So we'll see if there's repeats. Yeah, this is, yeah, Mark is just saying, this is just a topoisomerase, yeah. Yeah, Mark. This is one of the 20,000 proteins in a human. And that's why I said, if evolutionary biologists want to have a protracted debate with me, you're going to lose. Because I'm going to throw this stuff at you. I'm going to ask you every line of this. You tell me, how did this evolve? You got to explain it. Because I'm going to challenge you on statistics and cellular biology, why you think that's reasonable. If all you could say, well, it happened, you know, we have the peppered moths, I'm sorry, that's not going to cut it, buddy. You know,
right.
I'm sorry to be mean, but you know, they are destroying people's faith, and they're really hurting science by not appreciating the complexity. Because
that's the thing that I get. I'm like,
yeah.
it's the I think that a lot of people, I have actually never really done a lot as far as part of it's just I'm not good at evangelism in general, but you know, but mostly what I see is that science desperately needs a Christian point of view for it to keep on working correctly. And that's where I used to think that, you know, Christians needed science, but now the more that I see what's happening in science, science is really in need of what Christianity has to offer, because it's going in just, you can just see it going in the wrong direction with not understanding what they're seeing.
Yeah.
And
and
that's my worry.
And I could say that even more forcefully in some of the projects I'm working with Joe. And also, I mentioned it in the docket, the RScape, the AlphaFold, SciCalc, direct coupling analysis. We can actually move science forward if we look at the patterns of similarity and diversity as optimized to give us basically a user manual on how biology works. We can actually predict folds because of the patterns of similarity and diversity on the same protein across species. That's like voodoo. This is a great AI application, by the way, and at least a testable hypothesis, because I've been saying, why don't we just randomly mutate some of this in silicon? You know, we have the sequences, let's just random and see if you could still do full prediction. If you can't, that means a designer put that pattern there just so you could do fold predictions.
Mhm.
I mean, we're looking at the possibility that if you have a database of topoisomerases, for example, because we're talking about it, and you want to know how it folds, we can do it with X-ray crystallography. But let's say you didn't have X-ray crystallography, you wanted to know the shape. Well, you take then the topoisomerase of certain plants, you take some from yeast, some from humans, chimpanzees, rabbits. Then you can run AlphaFold, it'll give you the shape. But what if rabbits didn't exist? And you pulled out certain plants and suddenly you can't do fold prediction. You're like, oh my goodness. It's like the plant had to be created so we could do this. How does that make sense in an evolutionary paradigm that the plant is going to fill out a certain niche to help you do fold predictions for proteins, not just proteins, but for RNAs? And this is what Bill Dembski's steganography. And I found out, he said, well, actually, I have to give credit to David Chu. David Chu was Kirk Durston's PhD advisor. And Kirk and I and Joe are actually working on a project related to this with SciCalc. And so now the database is really going to be leveraged now. This is cutting edge ID, and it's just like, oh man, you know, we don't get to talk about this, and we put people to sleep, but the graduate students would appreciate this. So yeah, this is just topoisomerase. Yeah, this is just topoisomerase, and only one paralog at that. Oh, by the way, we haven't even talked about the alternative splices of topoisomerase. This is the main one. This is the layers of complexity.
Yeah.
So now, go
introns
on.
and extrons are just that's really where I think a lot of the cell specific magic is occurring.
Absolutely. And we're finding out the ALU elements, the transposon repeats are very important to triggering that. We've now elucidated the mechanism, and it actually deals with zDNA, which Michael Behe was a pioneer of. So you have the standard conformation, which we call Watson-Crick, but then you have 7 other ways. You can actually flip the DNA shape, and it's actually like flipping a bit. So you have information that's stored in 3 dimensions now that's regulatory. This is
So
mind-boggling. I mean,
so let me tell you about something that I read in the literature that was just amazing, and that is I forget exactly what the details were, but basically, if I think it was something with E. coli, and if it was being grown in a certain, I think this was when it's grown in arbutin. It will have, it's got coding sequences that digest arbutin, but there's no promoters for them. Okay, so they don't actually transcribe. But what happens is that if it's starving and in the presence of arbutin, the arbutin will actually change the way that the DNA is wrapped, or is I don't know, not necessarily folded, but the way the DNA is conformed, and it will turn it into a target site for an insertion sequence. And so I think it's IS5 is the insertion sequence that is so only if E. coli is starving and in the presence of arbutin, the arbutin will change the shape of the DNA to reveal a target site for an insertion sequence, which will insert a promoter sequence into the DNA that will then enable the transcription sequence to eat arbutin.
it's incredible.
Yeah,
That's incredible.
And it's like the thing that it needs to turn on is physically interacting with the DNA to change the DNA's conformation so that a insertion sequence I think it was arbutin. I could be wrong on which specific thing it was, but I think it was arbutin.
That's
But
incredible.
anyway, I just thought it was really cool.
Well, I think we're, if I could kind of finish this out, because we're not even finished with one database, but so it's a good thing you asked about domains and repeats. So we found it, and this is where we have the nuclear export signals, and I'll give a video on the export signals. And Mark O'Connell, if you're still there, all this complexity is surprisingly in just those amino acids, about 1,500 long. I mean, it's unbelievable something so compact could be so difficult and just do so much, because just the number of letters that, like, say, are in our side chat that tonight, that's way more than what's here, but it's taken the professional lifetimes of hundreds of people to elucidate how this thing works. It's just amazing. So Mr. Jetty's here. I want to, by the way, I'm sorry, I need to take a time out just to thank Mr. Jetty here. He's been a long time member of our community here, and he knows I've been doing stuff on nuclear physics and low energy nuclear reactions, and he's offered some really good objections to some of the ideas I put forward, and I want to acknowledge that because, you know, I don't want to give away bad data, and if there's something very questionable about the things that I say, I think it's important for those to be heard. And there are problems with some of the models and experiments that I proposed, and I'm hoping that they do get corrected. Mr. Jetty has, Mr. Jetty 89 has identified a lot of areas where for the radiometric problem to be solved for the young Earth creationists, particularly the solutions I've suggested, there's going to be a lot of work, and he's identified that, particularly the problems with the Coulomb barrier. So while he's on the other side of the planet, and while I get to see him live, I just wanted to give him full acknowledgement before we go back into biology here. So Mr. Jetty, if you're listening, thank you very much for your participation. So going back to this Uniprot knowledge base here, let me go back down. I lost my place. Yeah, so we were at the FASTA. Okay, so I went, oh, by the way, we mentioned AlphaFold. That's the, I guess that's, I wonder if that's the prediction algorithm that's so famous. It's not part of Uniprot. So, so we had the FASTA sequence here. And then somewhere here, see, it'll connect to, see, this is so detailed that even when we have, like in our sequencing labs, we think the sequencing's clean. They'll actually find variants or problems with the experiments. Then they have the alternative sequences. Here it is where they connect to the GenBank, which is the NIH's, the NCBI. That's the NCBI. The European counterpart is EMBL, and then they have others here. So if you want to look, and we'll actually just travel there briefly to show. And I'll just continue to the bottom. I hope I remember to go back there. Oh yeah, here are the 3D structure databases, which Joe he contributed to, at least he and his colleagues, his advisor at least, contributed to giving us 3D databases. I mean, this is, look at all the databases just for where you could see topoisomerase. But we're not even talking about the topoisomerase. We're just looking at the databases. We're not even talking about the details. I mean, this is a lifetime collection of experiments and data. And
Yep.
I feel for Joe for him to be a researcher here and to make an advancement in knowledge, he has to be knowledgeable about all the stuff that's published already. And
Right.
people, it's like, how can you study biology without studying evolution? It's like, how much evolution did I need to go through this database?
Yeah, I mean it's molecular microbiology, not history.
Okay, so I said I'm going to go to
GenBank.
yeah. So now let's go to GenBank. So you have to click GenBank, otherwise it's going to take you to the wrong one. And then now we can go to the Genomic Translation. I'll take this one. And this is where you can go to even more links in databases. And so this is where we have this. You can identify. This is another way of visually identifying the parts of the topoisomerase. It's not as, some tools are better than others, and there's a computational way to give you the parts of a protein visually, and that's CDART. And so when we go, do you know the name of your protein that you're of interest in?
Maybe let me. Well, let's see here. I don't want to get too distracted by specifics. Let's see here. It's AFGP12. It's one of them.
AFGP12. Okay, great.
Yeah.
So before we go there, I just wanted to show something here. This is a nice database, this CDART, because you can actually see if you lay out the amino acids from left to right, the proper term is from N terminus to C terminus. I call it left to right. You could see they block off the pieces and they give the position numbers of the little domains. So let me see if I could do this. Let me show another representation. PDP. Now I'm embarrassed. I should know the name of this. There is one that gave you nice little pictures. Well, just for lack of better, I'll just go with zinc finger. Sometimes you can get nice little domain pictures.
Now, one of the problems with some of the proteins and the one that I'm listing specifically is that it is actually intrinsically disordered.
Okay, we still may be able to work around that. So you can see kind of how they're able to colorize things, but CDART automatically does that. So why don't we just go to see if we can get lucky here. CDART. I mean, that's why you're here. Why don't we try it? Do a live demonstration, and if it works, then we can share it with your group. What else can we do on a Friday night, huh? All right. AF. Can you give the name?
It's AFGP12.
AFGP12. Right. Okay, this is looking for domains. All right, so that's the first thing. So why don't we go to Uniprot instead? Okay, so this is this is how we're going to stumble through this.
Yes.
Uniprot. Maybe we got lucky. AFGP12. I'd be surprised if there's a human that has it. Antifreeze glycoprotein in polar cod. Oh my goodness, it was the first entry. Let's see how much data we have on this. Okay, we have one, we don't have any recorded post-translational modifications. This is really different than topoisomerase. Either we haven't detected it or they don't exist. And so now here they have the disordered regions.
And if you look, do you see all those TAAs?
Mm-hmm.
So that's what gives it the antifreeze function, and that's the thing that and those are the repeats. Although there's two different sections of the protein that have the repeat of the TAA.
Wow, I'm sorry to disappoint you. That's the end of the Uniprot entry right there. You could see how it's so differently covered for a human, well-studied, protein like
Yeah. So
topoisomerase.
basically, what we need to do is find human proteins. Is that
It
what
helps.
we should do?
Well, I could tell you this. You just have to kind of. This is where serendipity plays in. You don't really know what you're going to find, and I can't generalize, but I would say that there are human proteins or anything that is a target of medical research, like insulin.
Mm-hmm.
You can bet it's. You can see how sparse this is as far as the. You compare to topoisomerase, how much more data topoisomerase had than this poor little fish antifreeze protein. So I don't even see where it says the point where it has repeats, domains and repeats.
It must come from a
Goodness.
different database that it's not in or something.
Yeah. And I wonder if they have a separate curator for each entry. That means they have a very large staff managing this, where people will volunteer. So the antifreeze glycoprotein.
So I'm looking up one now here that's a little bit more interesting because it's actually got a human variant. Search for runx-2. This might be something that people watching might be a little bit more interested in. Yeah. And so
Oh my goodness!
just
That is so cool. I think I saw that at the ENCODE conference that I was a part of in 2015.
So the runx-2, I don't know exactly what it does in humans. I mean, it generally controls the shape of the face. Specifically in dogs, it controls the bend of the nose. So this one does have that domains and repeats section you were talking about.
Wow.
Although
There it is. Yeah.
Although it doesn't actually list the repeat, which is weird.
And it may be one of these auto-generated sections that just going to call it there. If you happen to have a domain, it's going to be classified as domain and repeats, and then you don't have a repeat. That's why I suspect.
If you want to see the repeat, scroll down a little bit to the isoform. So note that section of Q's and that section of A's.
Oh yeah.
So basically, cool. I don't know what this repeat does in humans because it actually seems to affect different organisms different ways. But in dogs, it's the ratio of Q's to A's will determine the bend in a dog's nose.
This is so cool. Oh, greetings, David. David Neff.
I should probably head out. We've been at it for 2 hours, so I should probably get back to the family.
Okay, can I beg Krista to to grant me 5 more minutes because I want to show you the
Yeah,
localization peptides, and you've given me some ideas then how to present to your adaptive systems group.
cool.
And so this will just take me a moment to load this. If you wanted to have some words, while it's going to take me a little bit to get this going here. So
Yeah, yes,
if you had
Mark. With gene therapy, we can change a dog's nose. That's one of the questions. Yeah, so it's as I said, in other species, that ratio seems to do different things. Some species we're not actually sure exactly what it does. But one thing that's interesting about that is, see if I have the book around here. I don't know where I put it. There is a book called, it's by Darcy Thompson, called, I forget the name of it, but anyway, it was written a long time ago. But basically, what they showed is that that for a lot of different species, you can actually kind of convert one species into another simply by applying a coordinate transform. And anyway, I was that made me think that, I wonder how much of the differences between species are made by little ratios of repeats like this, so that you basically have, and basically all they're doing is doing coordinate transforms of different developmental activities. The book was on growth and form. And anyway,
I did not. I absolutely did not know that. So this has been a very educational show for me, and this is great because we're all so specialized in what we do. This is why it's great to have conferences like this. So let me see.
can I, yeah.
Well, go on.
Share real
I'm
quick.
trying to clean up my system here to get all this ready. So go on.
Let's see here. See my screen?
Yeah, here I can. There you go. If you wanted to highlight it.
So there. You have these two fish that are the same fish but differ by a coordinate transform. I'm going to stop screen sharing because it's repeating the audio in my ear.
Oh,
no,
sorry.
it's still working.
That was incredible. Can you show that one more time? And you might have to not talk while you show it. If I could just look at it one more time.
So yeah, so the book has all sorts of graphics like this, and they just show several different species where the different species are the same species, but just there's a coordinate transform that maps one to the other. And here's another example. That looks like two humans, but the book itself has a lot of more interesting ones, but I can't see the bidding.
This is I'm just going to kill this so you don't get the echo. What's the wiki entry that does this? That explains
DRC Thompson.
DRC Thompson. That I did not know that. You see, you're you study things at the organismal level. I'm down at the molecular level. We don't have conferences like this very often. That's great. Yeah, this could be fun. So actually, this is like a 4-minute video. This is one form of localization. And again, so the relevance to your work is to find out if you have localizations there, because then that probably wouldn't be a good place to be repeating because if you put the repeat in the wrong place, you're going to kill the localization. So that would be, all the more important. And unfortunately, they obviously didn't have the funding to do a characterization of the protein in this way, but there's probably a localization signal there. So let me play this here. This is, okay, I might have to mute both of us.
Reticulum, or ER, is the most extensive membrane system in eukaryotic cells. Proteins transported to the Golgi apparatus, endosomes, lysosomes, and the cell surface all must first enter the ER from the cytosol. As an mRNA molecule is translated into a protein, many ribosomes bind to it, forming a polyribosome. There are 2 separate populations of polyribosomes in the cytosol that share the same pool of ribosomal subunits. Free ribosomes are unattached to any membrane. Membrane-bound ribosomes become riveted to the ER membrane and translate proteins that are translocated into the ER. These membrane-bound ribosomes coat the surface of the ER, creating regions called rough endoplasmic reticulum. 2 kinds of proteins are moved from the cytosol to the ER. Water-soluble proteins completely cross the ER membrane and are released into the lumen, while transmembrane proteins only partially cross the ER and become embedded in the membrane. All these proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. The signal sequence is guided to the ER membrane with a signal recognition particle, or SRP, which binds the ER signal sequence in the new protein as it emerges from the ribosome. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane. The SRP is then released, passing the ribosome to a protein translocation channel in the ER membrane. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence functions to open the translocation channel. The signal peptide remains bound to the channel, while the rest of the protein chain is threaded through the membrane as a large loop. Once the protein has passed through the membrane, it is released into the ER lumen, after the signal sequence has been cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel into the membrane and rapidly degraded. It is thought that a protein serving as a plug then binds from the ER lumen to close the inactive channel. But not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as transmembrane proteins. For clarity's sake, the membrane-bound ribosome will be omitted to illustrate the translocation of transmembrane proteins into the ER membrane. In the simplest case, that of a transmembrane protein with a single membrane-spanning segment, the N-terminal signal sequence initiates translocation, just as for a soluble protein. But the transfer process is halted by an additional sequence of hydrophobic amino acids, a stop transfer sequence, further in the polypeptide chain. The stop transfer sequence is released laterally from the translocation channel and drifts into the plane of the lipid bilayer, where it forms a membrane-spanning segment that anchors the protein in the membrane. As a result, the translocated protein ends up as a transmembrane protein inserted in the membrane with a defined orientation. In some transmembrane proteins, An internal signal sequence is used to start the protein transfer, which continues until a stop transfer sequence is reached. The two hydrophobic sequences are then released into the bilayer, where they remain anchored. In complex multipass proteins, in which many hydrophobic regions span the bilayer, additional pairs of stop and start sequences come into play. One sequence reinitiates translocation further down the polypeptide chain, and the other stops translocation and causes polypeptide release, and so on, for subsequent starts and stops. Thus, multipass membrane proteins are stitched into the lipid bilayer as they are being synthesized, by a mechanism resembling a sewing machine.
Okay, that's ridiculous.
I know. But this is why it was nice. It's tying the Uniprot data to something like this, because when you read Uniprot, it'll talk about a translocon sequence, and it's just like amino. But then when you see what it means and how difficult it is to position it correctly, you're like, wow. Because it had to have foresight to start and stop. You have kind of a grammar there. You know, you need to have a, you know, you need the punctuation in terms of this, or like a left or right bracket. And that's just that's, and there's even more localization stuff going on. There are other localization, they call it localization peptides, and I had more videos. But and just like, okay, any sort of adaptation where you're changing the DNA sequence, it can't be random. You just see all the things that could go wrong, and this is kind of a nicer way. The reason I like this is a nicer way of approaching it, where it's not just vague and saying it's going to affect the fold. You can actually kind of see it in parts like this, where it's much more detailed and you can conceptualize it. It's not so abstract. So yeah, Paleologos. Yeah, see, Paleologos is he's a Todd Wood student here, and we've got all sorts of other people here, you know, undergrads and grad students. So this is cutting edge creationism and intelligent design. So I've learned a lot from you tonight, Johnny B, and I hope
Yeah, it's been
we'll
great.
be able to reciprocate here. So
Awesome.
well, I hope we're gonna. I'd like to say thanks to all those who viewed, joined us live, and also in via recording. And it's just great hanging out with you, Johnny B. We talked about calculus and all sorts of topics tonight, and be kind of scary if you weren't where you are, and we're, you know, across, you know, thousands of miles from me. We'd be hanging out. If you're local, it'd be kind of scary. You'd be hanging out all the time.
The world would not look the same.
Yeah. So I'll give you the last word, and maybe in the back channel, I'll see you backstage for just a little bit, and we can talk about how I could make the presentation to ERG. But with that, if you had some closing thoughts for our audience.
Well, I have to take my headphones off because I still got the echo. So yeah. So closing thoughts. I mean, it's just amazing what's in biology, and if any of you have the chance to become biologists, I highly encourage it, because it's an amazing area. And if you want to catch me online, I go to the blitheinstitute.org, or the blog that I write for a lot is mindmatters.ai. So you can catch me those places.
Alrighty, take care. God bless.
Thank you so much for having me on, and thank you all for staying for so long. It's been great.
And to our viewers, I echo that. Take care and God bless you. I'll talk to you later. Bye-bye.